My name is Lena, and I will be presenting the effects of Middle Eastern essential oils on E. coli. To begin, I would like to share a little bit of information about myself. My name is Lena, and I am currently a senior at Tunstall High School. And in the fall of 2023, I plan to attend the College of William and Mary to major in biology. I began research on this topic because of my concerns as a swimmer. Since we are in constant contact with the pool deck, we're exposed to all of its contaminations, which led me to research the most common bacteria found on pool surfaces, and it was E. coli. I wanted to test a substance that was both affordable and accessible to everyone, and I specifically used Middle Eastern essential oils because that's where my family and I are from. I also have an interest in healthcare, so it's interesting to learn about the different processes behind bacteria. E. coli can be spread so easily, so it is crucial to have new forms of treating the disease because of the severe symptoms it can cause. Before I conducted my experiment, I researched several studies to strengthen my understanding of the topic. Throughout my research, I have found that E. coli's membrane structure plays an important role when determining the susceptibility of the bacteria. It is a gram-negative bacteria, meaning it has a cell wall and proteins that gram-positive bacteria does not have. This makes the essential oils more resistant to the bacteria because of its complex nature. And when there is any alteration to the cell membrane, it disrupts the permeability of the cell membrane. Permeability controls which molecules, such as essential oils, may pass through, so the essential oils may have a higher chance of inhibiting the growth of the bacteria. Very few studies compare the most popularly used essential oils in the Middle East, which is why I decided to focus on frankincense, fennel, and thyme. Frankincense extract comes from a plant that grows along the Arabian Peninsula, and its components play a major role in how it will determine its inhibitory properties. Its major component is B-pinene, and in a study by Zaher et al., they found that B-pinene exhibited little to no antibacterial activity because of its inability to cause alterations to the cell membrane. It was tested against Staphylococcus aureus, and it failed to inhibit the growth of the bacteria. The next oil I tested was fennel extract. It comes from a plant that is grown in Asia Minor along the coast of the Mediterranean Sea. It is mainly composed of trans-ethanol, carbapenem, and thymol. Swami et al. Found that trans-ethanol was responsible for affecting the cell's energy system. When the cell loses energy, it will no longer be able to function, meaning the essential oils will be able to inhibit the bacteria growth. Another study said that thymol and carbapenem were responsible for disrupting both the outer and inner cell membrane, allowing the essential oils to come and pass through. The study also revealed that fennel extract was able to inhibit another gram-negative bacteria called Shigella dysenteriae. The last essential oil I tested was thyme, which is found in the Levant region of the Middle East. It is heavily composed of carbapenem, thymol, and eugenol, which are all linked to antibacterial activity, and it was found to inhibit Staphylococcus aureus that was isolated from a vegetable. The question I formed to conduct my research was which Middle Eastern essential oil will exhibit the largest zone of inhibition when presented against E. coli. After forming my question, the hypothesis I created to show the relationship between my variables was if thyme is presented against E. coli, then it will exhibit the largest zone of inhibition. My null hypothesis was if all three essential oils were compared to one another, then there would be no difference in how they perform against E. coli. The independent variable was the type of essential oil, which was either frankincense, fennel, or thyme. The dependent variable was the growth of the bacteria. And to compare how the essential oils would affect the growth of the bacteria, a petri dish of just bacteria was used for the control. In order to ensure the validity of the experiment, the nutrient agar and the temperature remained constant for each treatment. I began my experiment by obtaining the essential oils, and then I then gathered the petri dishes, the bacteria, and swabs to begin plating the dishes. I had a randomized sample of 40 petri dishes, and they were separated by 10 groups of each essential oil. In the picture on the left, I obtained a sample of the bacteria, which was the sample population, and began swabbing it on the petri dish side to side while rotating the disc to create a uniform layer of bacteria. While I was swabbing the bacteria, the essential oils were saturating in the beaker, which is seen in the top right photo with the discs. In the bottom right photo, with sterilized forceps, I placed one essential oil infused disc onto the petri dishes and then closed the dish. When all of my treatments were done, I placed the petri dishes into the incubator for 48 hours at 37 degrees Celsius. Once my treatments were done incubating, I recorded the zones of inhibition in millimeters and then transferred the data into an Excel spreadsheet. I then conducted a statistical analysis. The statistical test I used to perform for my data was a one-way AND At the significance level of 0.05, the null hypothesis was rejected with a p-value less than 0.001. To visually display my data, I used a bar graph to represent the results of each essential oil. The graph depicts the average zone of inhibition in millimeters for the essential oils frankincense, fennel, and thyme. As thyme had an average zone of inhibition of 26.45 millimeters, it was the largest inhibitor, while fennel was the next largest inhibitor with a value of 6.95 millimeters. The graph shows how frankincense did not inhibit the bacteria because there was no zone of inhibition. The first petri dish contains frankincense-infused disc, and it had complete coverage of bacteria, meaning no E. coli was inhibited. The second petri dish contained a fennel-infused disc, and it had a slight zone of inhibition where no bacteria was growing on the dish. The last petri dish with thyme had total inhibition, so it completely inhibited the E. coli. Based on my experiment, it can be said at the 0.05 level of significance, there is sufficient evidence to conclude that the essential oil thyme is the largest inhibitor against E. coli. Thyme's large performance may be due to how it was mainly composed of carvacrol and thymol, which were linked to immense antibacterial activity. Frankincense's lack of inhibition may be due to it was mainly composed of monoterpenes, which is a chemical compound that was found to not exhibit very much antibacterial activity. The results of my study concluded that thyme and fennel may be used as natural inhibitors. It is important to have more treatments available because of the risks associated with E. coli. There is also an increasing risk of antibiotics to the bacteria, so the addition of essential oils may be necessary to combat the disease. And the essential oils thyme and fennel may be used as a topical treatment to treat the infection. And they can effectively inhibit the bacteria, so they are a potential candidate for being used as a preservative in foods. The essential oils were commercially obtained, which may have altered the results because of possible synthetic additives. The study would have been more appropriate if the essential oils were naturally extracted, but because of time and limited resources, I was unable to experiment with the oils in their purest form. And the essential oils were not purchased from the region they derived from, which could have also made the results less accurate. For more accurate results, there should have been a larger sample size, and because it would have been closer to the actual population. Future research should expand on the types of essential oils used while increasing the sample size to obtain more accurate results. Throughout this process, I have learned the importance of time management skills. While learning to effectively manage my time, it was crucial to have a positive and determined mindset because it made the experiment more enjoyable and efficient. And after reading many studies about how dangerous and common E. coli can be, it has encouraged me to take more precautions to prevent myself from obtaining the disease. I'd like to thank Ms. Long for assisting us in the lab and answering any questions we had during this long research process. I'd also like to thank Ms. Dillon for helping me in the lab. And lastly, I want to thank Dr. Jones and Dr. Pocock for allowing me to carry out my research. At this time, I'll take any questions. Mr. Taylor.
Yes. What tool did you use to measure your zone of inhibition on the petri dishes?
Mr. Taylor asked what tool I used to measure my zone of inhibition on the petri dishes, and I used a rule and numbers.
And was that a radii measurement, diameter?
Yes, I measured from the middle of the disc to the end of the petri dish. Ms. Moore.
Yes, so I saw that you had the randomized sample of 40. Does that mean you had 40 of each of the independent, the essential oil, and 40 of your control? Or you randomly selected which petri dish was going to be a control versus the oil?
Ms. Martin is asking about the randomization of the petri dishes, and it was a total sample size of 40, so they were separated by 10 groups, so 10 control, 10 frankincense, 10 fennel, and 10 thyme, and I used a random number data to randomly assign the petri dishes to a group.
Okay, thank you so much.
Dr. South.
How did you decide to grow your bacteria in 37 degrees Celsius?
Dr. South asked how I decided to randomly grow my bacteria in the 37 degrees Celsius, and all of my studies used it at 37 degrees Celsius, so that was the optimal temperature for bacteria to grow.
Okay, thank you.
And the brand of oils that you used, is there a specific reason you focused on those?
Ms. Ashworth asked about the specific type of essential oil, like the brand. I just used, are you asking why it was the Middle East or?
No, why that particular brand?
Okay.
Yeah.
I just used that because it was the most accessible and it was just easy to order, and it all had the same essential oil with the brand. So it was.
It's a good company, that's why I didn't know if you did research to find out anything about the purities. Good, thank you.